Hi, thanks for stopping by today. My name is Heather Kosakowski, and today I'm going to talk to you about face selectivity in the human infant brain. In our daily lives, humans encounter a variety of faces, such as this one. When you see a face like this, you might notice that this is a girl with pale skin and freckles. She has blue eyes, and if you know her, you'd know that this is my daughter, as opposed to another similar-looking 11-year-old girl. Face perception and identity are processes thought to be supported by the Lateral-Occipital Cortex and the Ventral-Temporal Cortex. Beyond knowing the identity of this face, you might notice that her facial expression and whether or not she's already engaged in a social interaction. Perhaps here you notice she's smiling, maybe even laughing at the person taking her picture. Face dynamics, such as those involved in facial expression and social interaction perception, are functions thought to be supported by the superior temporal sulcus. But perhaps the most important aspects of a face are the features that help you determine if this is someone you want to engage with. Would interacting with her be enjoyable? Is she looking at me? And does she share a desire to engage socially? What would we talk about? If you know her, you might know she likes Avenger movies, math, and is in the 6th grade. Information that can help you decide which topics to discuss and what kind of social partner she might be. These kinds of functions are thought to be supported by the medial prefrontal cortex. How does the brain develop to support these functions? A commonly accepted theory is that brain function develops posterior to anterior. This idea is supported by data that show the brain develops myelin along the posterior to anterior axis. This predicts that posterior face-selective regions, such as the one in the lateral-occipital cortex, would develop first, followed by the ventral-temporal cortex, then the superior temporal sulcus, with functional specialization in the medial prefrontal cortex developing last. It also predicts that higher order social functions, such as social partner identification and social interaction perception, would emerge later in development than face-specific perceptual functions, such as processing face parts and facial identity. So we used fMRI to see which of these different regions we would find in awake infants. That is, do infants have face-selective responses in lateral-occipital cortex, ventral-temporal cortex, the superior temporal sulcus, or the MPFC? For those of you that are not familiar with infant fMRI, we first swaddle the infant and place them in a special infant coil. We use a sinusoidal EPI sequence that is quiet and safe for infants' ears. An adult goes into the scanner with the infant, while babies watch movies in a mirror over their eyes. As you can see, this baby is moving quite a bit. We improved data collection techniques by first designing a set of scanner-safe headphones that allowed us to use a higher resolution EPI sequence without harming infants' ears. Then, in collaboration with Forrest Kell, we designed a new infant head coil that maximizes infant comfort and has headphone cutouts. The frontal coil then flexibly closes around the infant's head and is held in place by inflatable pillows. While in the scanner, babies watched movies of faces, bodies, objects, and scenes. To hold babies' attention, we also had a baseline block that consisted of colorful abstract videos. The movies played continuously as long as the baby was content and awake. We recruited 86 infants and collected a total of about 47 hours of data from 70 of these infants. As you might remember from the videos, infants move a lot during scanning. As such, runs were truncated into subruns by removing high motion segments. This scrubbing left us with about 16 hours of low motion data from 49 subjects. To be included in the functional region of interest analysis, infants had to have at least 2 subruns with at least 4.8 minutes of data in each subrun, leaving us with a total of 13 hours of data from 30 subjects between the ages of 2.5 and 9.4 months. For 16 of these subjects, we were able to use the higher quality data collected with the new coil. All right, so after all of that work, what did we find? We first looked in the ventral-temporal cortex, a region that is famously known for the fusiform face area. We compared faces to objects, and in a group random effects analysis, found the face response with both the old coil and the new coil datasets. Then, we selected voxels that had a greater response to faces than objects in the ventral-temporal cortex, and assessed the response to faces, bodies, objects, and scenes in independent data. Here I will show you the response magnitude on the y-axis. The response to faces is here in purple. As you can see, it is significantly greater than the response to bodies in pink, objects in teal, and scenes in green. So the face response in infant ventral-temporal cortex is face selective. What about the lateral-occipital cortex? This region is earlier in the neural hierarchy than the ventral-temporal cortex and is where the occipital face area is found in adults. In the group random effects analysis, we did not observe a face response in the old coil datasets. We did, however, find a face response with the new coil data that was more dorsal than what we would expect for the OFA. In the functional ROI analysis, the face response was not statistically different than either the body or the object response, and only marginally greater than the scene response. Thus, infant lateral occipital cortex, we did not observe a face selective response. All right, now what about the superior temporal sulcus, a higher order social perception region? Here we found face activations in the STS for both the old coil and the new coil datasets. In the functional ROI analysis, the face response was significantly greater than the response to all other conditions. So yes, the infant STS is face selective. Finally, we looked at the face response in infant MPFC, the most anterior region. In group random effects analyses, there was an MPFC face response for both the old coil and the new coil datasets. In independent data, the face response was greater than the response to all other conditions. So infant MPFC is also face selective. All right, so far I've told you that we did not observe face selective responses in the lateral occipital cortex of 3 to 9 month old infants. But in the same group of infants, we found face selective responses in the ventral-temporal cortex, the superior temporal sulcus, and medial prefrontal cortex. So in this group of infants, we found face selectivity in frontal regions of the infant brain at the same time in development as the more posterior face selective response in ventral-temporal cortex. But perhaps we could still find evidence that the ventral face response emerges prior to the STS or MPFC face responses. All right, we found no evidence that posterior regions develop face selective responses prior to anterior regions. But can we find evidence of age-related change that would suggest the ventral region is face selective prior to anterior regions? To answer this question, we looked at the difference between the face and object response as a function of age in each region. On this plot, I'll show you the age on the x-axis and the face minus object response on the y-axis. In lateral occipital cortex, we found that the magnitude response difference between faces and objects was bigger in older infants than in younger infants. The same is true in ventral-temporal cortex, STS, and MPFC. So, in this group of infants, we found no evidence that posterior regions develop selective responses prior to anterior regions. In fact, we observed face selective responses in ventral-temporal cortex, STS, and MPFC at the same point in development. All right, so I've told you about infants' face responses in lateral and ventral-temporal cortices, STS, and MPFC. But using reverse inference, I also associated infants' neural response to faces to functions we know to be true in the adult brain. Specifically, I said the face response in lateral occipital cortex is perceptual and has been associated with face part perception. The face response in ventral-temporal Ventral-Temporal Cortex is thought to be associated with face identity, while the STS is thought to support processes such as facial expression and social interaction perception, and MPFC is thought to support social partner selection. Do we have evidence in this data set to suggest that is also true of the infant brain? To answer this question, we looked at how each face ROI responded to 3 different face conditions: forward neutral faces, averted neutral faces, and forward expressive faces compared to objects. In a region that processes face parts, you might expect that frontal views, in which eyes, nose, and mouth are in canonical locations with typical shapes, would have a high response, whereas faces with a side view, and thus a different shape for the eyes, nose, and mouth than what is typically perceived, would be lower and perhaps similar to objects. Conversely, a response in a region that processes face identity should be able to recognize the identity of any face, regardless of face orientation. That is, face responses should generalize to all viewpoints and be greater than objects. A social interaction perception region will respond more to faces that are clearly engaged in a social interaction or that have more animated expressions. A region like this should respond to all faces more than objects, but slightly more to dynamic, socially engaging faces. Whereas a region involved in social partner selection will respond to faces that look like they are ready to engage in a meaningful social interaction with me. A face response that is specific to social partner selection would respond to all faces with a direct gaze and slightly less to faces with an averted gaze. All right, so the infant lateral face response preferred front faces, and the response to averted faces was similar to the response to objects, a response profile that may be consistent with processing face parts. In ventral-temporal cortex, the face response generalized across viewpoints, a response profile that is consistent with the functional profile of adult face responses in the ventral-temporal cortex. Interestingly, the STS face response preferred dynamic, expressive faces, similar to the adult function of social interaction perception, and the MPFC had a high response to all faces, but had a slight preference for faces with a direct gaze. This is consistent with the role MPFC has in social partner selection. Research using fNIRS with infants supports this high-level reverse inference. Using fNIRS, researchers have found that infant STS responds to dynamic faces, point light displays of face and biological motion, and shapes that appear animate. Similarly, infant fNIRS research has shown that the MPFC responds more to faces with a direct gaze, familiar faces, and faces associated with infant-directed speech, smiling, or even playing peekaboo. Taken together, these results suggest that social regions of the infant brain, such as STS and MPFC, already have distinct functional profiles that are similar to adults. All right, so today I told you the face response we observed in infant lateral-occipital cortex was not face selective and responded most to forward faces, a response that is consistent with the role of lateral-occipital cortex in adults. We did observe a face-selective response in infant ventral-temporal cortex, and the face response generalized to all views, a response profile that is consistent with the FFA, a face-selective region in the adult ventral-temporal cortex. In infant STS, we also observed a face-selective response, but here, the region preferred dynamic, expressive faces compared to neutral faces, a response that is consistent with both the functional profile of adult STS and previous infant research using fNIRS. Finally, infant MPFC is also face-selective, but here, the response to any face is high, with a slight preference for faces with a direct gaze, a functional profile that is also consistent with the proposed role of MPFC in social partner selection. Additionally, there was an effective age in all regions such that older babies had a more selective response than younger babies. Taken together, we find no evidence for posterior to anterior functional development in the infant brain. Rather, we find face-selective responses in infant MPFC and STS at the same time in development as ventral-temporal cortex. With that, I'd like to say thank you. Thank you for your attention, and thank you to my collaborators and all of the people that helped me, all of the babies and families that participated, and my funders.